Right, so we were actually saying that for phosphoric acid, we see that it has 3 equivalent points. We saw the pKa1 being at 2.15, pKa2 being at 7.2, pKa3 being at 12.4. So this is a triprotic, okay, acid. Okay, we can be able to deduce that this is a triprotic acid because we are able to see 3 inflection points. We have this inflection point, okay, we have this region, we have this region with the inflection point on those areas. So 3 inflection points, meaning this is a triprotic or dibasic acid. Okay, but we can also say that this phosphoric acid has 3 buffering regions. Okay, it is able to act as a buffer in the pH range, in the pH around 2.15. It can also act as a buffer in the pH range of 7.2 and also at the pH of around 12.4. Okay, it can act as a buffer because we know that this is an acid and this is a conjugate base. So if you have this acid and the conjugate base, then it can act as a buffer at that pH. If you have this acid whose conjugate base is this one, it will act as a buffer at 7.2. If you have this acid and its conjugate base, it will then act as an acid at a pH of 12.4. Okay, so we can say that the phosphate ion can act as a buffer. Okay, phosphate is found in the blood and the body, so we can actually say the phosphate ion is a buffer that we can utilize in our body. Okay, the phosphate is one of the buffers that we can utilize in our body. Remember, the pH of our blood is around 7.35 to 7.45. Okay, so if you look at the pKa2 of the phosphoric acid, it is about 7.2. Okay, remember we said that a buffer can act as a buffer if it is at 1 plus pK or 1 minus pKa. So this falls in that range. So this can act as a buffer in the human blood. Okay, the human blood has a pH of 7.35 to 7.45, which we sometimes call the physiological pH. Okay, the physiological pH is a pH of a human blood. Okay, right. So that's one example of the pH that can be utilized in our bodies, so to say, in humans. Okay. Right. The other buffer that can be utilized in the body include the bicarbonate buffer. You know, in the blood, hydrogen ions can react with the bicarbonate ions, and in the process they form an acid, carbonic acid. Okay, but this carbonic acid also can dissociate, producing water and carbon dioxide, which can then be emitted out. Okay, right. So we are saying that this is a base. We'd say this is a base. Okay, this is an acid. How do we know this is an acid? We said that an acid has more hydrogens than the base. Compare this and this one. This has 2 hydrogens, this has 1 hydrogen. So the bicarbonate qualifies to be a base. The carbonic acid qualifies to be an acid. Okay, So, however, we know that the carbonic acid can equilibrate with carbon dioxide. So the concentration of carbonic acid can be given by the partial pressure of carbon dioxide in our blood. Okay? So normally the concentration of bicarbonate ions is given as 0.03 times partial pressure of carbon dioxide. Okay, that's a correlation that is between the concentration of bicarbonate ions to the partial pressure of carbon dioxide in the human blood. Therefore, if you go to the Henderson-Hasselbalch equation, you have pH is equal to pKa plus log, okay, a base, we say base over concentration of the acid. Okay, so we can then say pH is equal to pKa of the bicarbonate ion is 6.1. Okay, that's the pKa of bicarbonate ion. See, 6.1. Okay, plus log of the base. In this case, the base is the bicarbonate ions. So the concentration of bicarbonate ions over the acid. The acid is carbonic acid. But carbonic acid can be related to the partial pressure of carbon dioxide. So if we use the partial pressure of carbon dioxide, then we say 0.03 times partial pressure of carbon dioxide. Okay, that will give us the pH of the human blood. Okay, that will give us the pH of the human blood. Okay, under ordinary condition, okay, you and me, when we are okay, the normal concentration of bicarbonate ions, okay, the normal concentration of bicarbonate ion is about 24 milli equivalent. 24 milli equivalent. And then the partial pressure, the partial pressure of carbon dioxide normally is about 40 millimeters of mercury. Okay, so if you slot in that, so we are going to have pH is equal to 6.1 plus log of concentration of bicarbonate ions, which is 24. Okay, then 0.03 times the partial pressure of carbon dioxide, which is 40. Okay. If you do your calculations well, you are going to have 6.1 plus 24 divided by this. Do your calculation, and then you find the log. I'm sure it will give you 1.2. 1.2, somewhere there. Okay, so the pH of the human blood then will be 7.3, which is within the range, the physiological range of 7.35 to 7.45. That is the physiological pH. Okay, that's the physiological pH. So this is another buffer that can be used in the human body. Okay, so in the human body we've talked about two buffers. The phosphate buffer, whose pH is around 7.2. We have also talked about the bicarbonate buffer, whose pKa is around 6.1. Okay, hemoglobin can also be used as a buffer. Okay, hemoglobin, another amino acid, but particularly hemoglobin, can be used as a buffer using the histidine residues. Okay, using the histidine residues. Right. So ideally, these are buffers that can be used in the body, and they will tend to maintain the pH of human blood at a constant level of 7.35 to 7.45. Right. So in the lab, we can use acetic acid as a buffer, acetate buffer. We can use the phosphate buffer. Okay. Apart from these, there are other synthetic buffers that have been synthesized. Okay. Each having a particular pKa value, different pKa values. One of the best synthetic buffers that we can think of is the Tris buffer. The Tris buffer. Okay, Tris buffer, which if you want, you can write it this way. Okay, this is CH2 hydroxide. This is the amino group, which is protonated, and CH2 hydroxide. Okay. So we can say this is tris hydroxy methyl lamino methane. That is it. In short, we say the tris buffer. Okay. What you observe is that the amino group is protonated. So this can undergo dissociation, releasing a proton from the amino group. Once a proton is released, what you have is so you have the amino group without it, which is without hydrogen. Okay. So the tris, which is protonated, can be deprotonated to form your tris. So this is tris, if you want tris, we can say it is positively charged. And this is still tris, which we can say it is without charge. We can say tris 0. Okay, full name is tris hydroxy methylamino methane. Okay, this has a pKa value of about 8.1. Okay. Right, this is a synthetic one, so we can mimic the pH range of the human blood by using the tris, because we are saying that it's 8.1. Human blood is 7.35 to 7.45. Okay, if you add 1 or you subtract 1, the buffer can still work. So 7.45 plus 1, that will be 8.45. So this will be a good buffer to mimic the human blood buffer. Okay, so basically if you want to find the pH of a reaction or the environment that you are working with, if you are using the tris buffer, it will be pH is equal to pKa plus log of the base. Okay, the base is the one that has less hydrogen, so it's this one, tris 0, concentration of tris 0 over the acid. The acid is the one that has more protons, so tris plus. Okay. Then if you have been given the concentration of the tris which is not protonated and the tris which is protonated, you can insert them there and set the pKa value. The pKa value is 8.1. Now there are other many synthetic buffers. I've just given you one. Okay, so let's look at one question or two questions. Okay. The question says blood plasma contains a total of carbonate, total carbonate pool of 2.52 times 10 to the power minus 2 molar. Okay, then calculate, okay, A, what is the bicarbonate carbon dioxide ratio, okay, then B, what is the concentration, of each buffer component present at pH equal to 7.4. It's not 4 but 7.4. So remember what gives us a ratio of the components. What gives us a ratio of the component is the Henderson-Hasselbalch equation. Okay, so we need to write Henderson-Hasselbalch equation. So pH is equal to pKa plus log of the base. The base, remember we said it is the bicarbonate ions. The acid is the carbon dioxide. Okay. We are given the pH. Okay, we are given the pH. We are also given the pKa. I gave you pKa to be 6.1. Okay, so pKa. So then we need to insert, since we have been given the pH is 7.4, pKa is 6.1 plus a log of concentration of the bicarbonate ions over carbon dioxide. Okay, so 7.1 minus 6.1, this comes this side, so it becomes minus log bicarbonate ions over carbon dioxide. Okay, so if you subtract that, what will be 1.3 is equal to log of concentration of bicarbonate ions over carbon dioxide. Okay, so then you can find the antilog. So you can proceed on and find the antilog of 1.3 will be equal to the ratio of bicarbonate over carbon dioxide. Okay, I have no calculator here, so you can calculate that. That will be the ratio. Okay, then second question, what is the concentration of each buffer component? Right. Okay. What have we been given? We were given the statement read, blood plasma contains a total carbonate pool of 2.52 times 10 to, so that's a total. So what you can do is that after you found the ratio, okay, after you found the ratio, I don't have a calculator. There is no calculator. I don't have a scientific calculator. So if you calculate the ratio, then you can say that just for argument's sake, okay, for argument's sake, if you say it's 20 to 1, okay, it's not the ratio, calculate the ratio, but if it's 20 to 1, so concentration of bicarbonate over concentration of carbon dioxide, then you can do the more ratios. So you can say that the concentration of bicarbonate ions is equal to, what's the component of bicarbonate? 20. What's the total? It's 20 plus 1 times the total concentration, which we are given as 2.52 times 10 to the power minus 2 moles per cubic decimeter. Similarly, you can say concentration of carbon dioxide will be equal to, what's the component of carbon dioxide is 1 over 20 plus 1 times 2.52 times 10 to the power minus 2 moles per cubic decimeter. Okay. And so on and so forth. Okay. So having done that, I guess I'll stop here and give you exercise, or is it assignment? Okay, so assignment. Question 1. Calculate the pH of a buffer composed of 0.060 moles sodium dihydrogen phosphate and 0.040 moles of disodium hydrogen phosphate in a total volume of 1.0 liters of aqueous solution. Given that you are given the equations, okay, plus water gives you that pKa 2.15. Then we are given that plus water gives you that, which is minus, which is 2 minus, plus water, hydroxinium ions, pKa, you are given as 7.2. Then you are given that is minus, is 2 minus, plus water gives you that plus phosphate ion. pKa equal to 2.4. Okay, so that's your question. Okay, I can wrap. Then the other one, an enzyme catalyzed. Enzyme catalyzed reaction was carried out in 0.2 molar trace buffer. Okay, pH equal to 7.8. Okay. Okay. As a result, the result of the reaction, result of the reaction, 0.03 molar of hydrogen ions was produced. Okay. Question A. Question 2. Okay, so what was the ratio? What was the ratio of trace star cheesy acid to trace not cheesy base? Okay, and hence the concentration, and hence calculate the concentration of these at the start of the reaction. Okay, B, what were the concentration? What were the concentration of trace star to trace not at the end of the reaction? C, what was the pH? What was the pH? What was the pH at the end of the reaction?